All right, in 25.3, we're going to talk about identification using DNA methods, one of my favorite things. We're going to talk again about the polymerase chain reaction. We'll talk about a modified version of it called qRT-PCR, quantitative real-time or reverse transcriptase PCR. We'll talk about the pros and cons of this and a few other related techniques. Okay, so PCR, polymerase chain reaction. This is one of the most widely used, what we call molecular methods in the clinical lab. It is cheap, it is fast, it is super sensitive, and it is very accurate. PCR is super useful for identifying viruses because, as we know, viruses need other cells to grow. So you cannot grow viruses on a plate. That means detecting them is difficult, and PCR can help us with that. Remember, in PCR, we use little DNA primers. Those match to sequences. And if they match to the sequence, then we get multiple copies of these things made. And that helps us detect it. So we make different primers for different pathogens. If we add them and we get copies made, that means the pathogen was present. You can even do this with many different sets of primers to identify specific genes from a pathogen to get like down to the strain level. What type of strain of bacteria is this? So this whole technique, PCR, is really great for things that are hard to grow, that are slow to grow, things like Mycobacterium tuberculosis and stuff like that, that don't grow well on plates. So the key to the PCR reaction, remember, is the primers, the short little pieces that will match to the DNA sequence. If they match, we get copies, and we can view that on an agarose gel, right? We could see that little band of DNA if there were copies made, no band if there's no copies. But there are new technologies like real-time PCR that can skip this gel step and just do it all in one machine. So we can use a machine that can detect the number of copies that have been made. So PCR is great because if we do it enough, we can amplify starting even from one molecule. So it is super sensitive. We can detect even a very small amount of pathogens in there. All right, so let's talk about what's going on, right? Here is a PCR machine, very like the one we have. In here are these little wells, and the PCR tubes go in there, and then the cycling of the reaction occurs. Remember, each PCR reaction has a DNA sample. That might be the microbe that we're testing for. 2 primers that we specifically put in there. Nucleotides A, G, T, and C. The special Taq polymerase that can withstand the high temperature. A buffer, and we put it all in a tube, and then we cycle it. So remember, the first cycle, we heat it up to 95, which denatures the DNA, splits it apart by breaking those hydrogen bonds. The second step, we lower the temperature a bit to anneal or attach the primers. If they're going to match, they will match in this step. Then we raise it to 72 and we extend, making a new copy. So we started with 1 double-stranded DNA. Now we have 2 double-stranded DNAs. We repeat these cycles over and over again, and if our primers match, we get lots of copies being made. So let's look at an example of this. 55-year-old man comes in complaining of a headache, high fever, and neck stiffness. Maybe perhaps meningitis. We'll see. He's confused, disoriented. Also complains of muscle weakness, which is a bit more than we've heard in meningitis before. He mentions that he received a large number of mosquito bites 2 weeks beforehand. So we're going to take cerebrospinal fluid and blood and send it to the lab. But we were suspecting meningitis. The next day, we find no growth, right? No indication of a bacterial infection. So where do we go from here? Well, now we start looking at viral infections, right? If no bacteria grew on a plate, could be viruses that are the cause. So we are going to use a process called quantitative reverse transcription PCR to test for various viruses in there. And each one of these has a specific primer pair, so we set up a whole bunch of reactions for a bunch of common viral infections in that particular region. In this case, we find it is West Nile virus. West Nile virus can infect humans, horses, and some other organisms. And it is the leading mosquito-borne virus and disease, really, in the United States. We don't have malaria here, thankfully, but West Nile is common in the United States. Luckily, most people don't develop any illness from it, but 1 in every 150 infected individuals does develop a severe infection. It could potentially be fatal. To culture West Nile virus, right, to grow it, you would need to do tissue culture where you have human cells in a petri dish and introduce the viral sample. And to be honest, most diagnostic labs are not set up to do that. It's too expensive, it's too time consuming, it just doesn't work well. So the PCR technique worked very well. It's very rapid, identifies it in a matter of hours. We could also theoretically measure antibodies in the cerebrospinal fluid, but this isn't as good of evidence as that PCR positive. So we tested for viral RNA, but there's a problem here. PCR works on DNA, not RNA. So let's talk about this. We've mentioned this with COVID before. What we have to do is what's called reverse transcription PCR, which takes RNA and turns it into DNA, and then we can use that DNA to do PCR. And the quantitative part in here means we can actually count how many copies get made. I'll show you how that works in a moment. But let's look at reverse transcription. So reverse transcriptase, you probably heard about this when we talked about HIV. It's an enzyme that takes RNA and makes a DNA copy of it. So it's going backwards from the process of transcription. So here we have viral mRNA. We add reverse transcriptase and it works backwards, making a DNA copy. We call this cDNA, complementary DNA. And we now have DNA that we can work with to go into PCR. So we got to take the RNA, take it out, turn it into DNA using reverse transcriptase. Then we're going to do a special PCR reaction that uses fluorescent dyes. So we do a PCR reaction, and Taq polymerase comes in, and there's a fluorescent molecule here that gets disrupted when Taq polymerase comes along. The amount of fluorescence that is occurring can be measured by a very sensitive and expensive type of camera to basically count how many copies of DNA have been made at each step. This gives us an estimate of how much we started with. If we work backwards, we can figure out how much we started with. So let's see an example of what the results actually look like. Okay, so remember, PCR, start with 1 molecule, end up with 2 after the first cycle, 4, 8, 16, so on and so forth. There is kind of an upper limit. Things get very crowded at the high limit. So what is this quantitative PCR doing? Well, it's looking for when we start going into basically an exponential phase of growth here. And the early stages, it can't detect it. But as it goes along, the fluorescence starts to get detected. So we have cycle number down here and basically how brightly it's glowing or how many copies are in there on the y-axis. So if we look at this, cycle numbers down here, try to interpret this and think about which patient started with more virus. We have patient 1 versus patient 2. Which one started with more virus to begin with? Go and pause the video. Think about that for a moment. Okay, so if we look at this, patient 1 started going into this exponential phase at cycle 14 or 15, I'm going to say. Whereas patient 2, it took all the way up till cycle 25 or 26 for them to go into that exponential phase. what's that mean about how much DNA they started with, or viral RNA they started with? Well, if it takes less time to get to a high number, that means we actually started to begin with a much higher amount. So this patient, because they took less cycles, had more viral RNA in their sample than patient 2, which took longer to go up to this high level. So we can use quantitative RT-PCR to actually calculate how much virus is in a patient, which can be quite useful, particularly in like HIV, where we really want to make sure that there is no detectable virus in a patient to prevent transmission and other things. All right, so we've talked about PCR, right? I just want to hit it one last time. It's my favorite thing in the world. qRT-PCR is a modified version of that. This all works really well for detecting viruses, particularly the quantity of virus in a sample. All right, in the next section, we will talk about using antibodies to detect microbes.